These are the notes about DNA replication. Today we're going to describe how DNA replicates, and you need to make sure you understand the term semi-conservative replication. Replication is simply the process by which DNA copies itself. When we talk about complementary strands of DNA, we're talking about the fact that they go together well, not the kind of complementary that's the free breakfast at the Holiday Inn. We're talking about that A complements T, that they go well together, that G complements C. DNA is double-stranded, meaning this is one strand on the left-hand side, this is another strand. DNA has two steps. The first is the separation of the strands. And in essence, the DNA unzips. And if you'll hang on a moment, I'll give you a visual. The second step is we add nucleotides to each of the unzipped sides. The result is two identical copies of DNA. And here's where the semi-conservative part comes in. Each new strand has half of the original strand. If that sounds weird, hang on, I'll show you a picture. So let's start on the left-hand side. Here are the complementary strands of DNA. The first step is to unzip them. So we have split them apart, essentially. The second step is adding new nucleotide bases. You see that they're adding new nucleotide bases here. The end result is two identical DNA molecules. The semi-conservative part comes in where each of the two new molecules have half of the original DNA, shown in dark blue, and a new strand, shown in light blue. Excuse me. Now this was a very simple diagram. Here's a more realistic diagram. We know that the DNA is a double helix. So there is an enzyme called DNA helicase that sits right here, and it will unzip the DNA. There's another enzyme, DNA polymerase, that will sit on each single strand and add nucleotides. The new nucleotides are shown with a red backbone in this photograph. There is a video about DNA replication. It's available on my YouTube channel. Feel free to go check that out. In your foldable, you will draw one or the other of these replication models on the far right-hand flap on the inside of your foldable. Now, there are a few errors that can happen during replication. I asked the class to guess how often you think these errors occur. It's actually 1 per billion nucleotides. I think this is pretty good. When errors occur, they're called mutations. Now this is what I want you to write down at the very bottom of that right-hand flap, underneath the picture of replication that you drew. Mutations are simply errors during replication of DNA. So this second bullet point is really all you need to write down. But let's go ahead and look at what if an error occurs. There are several different proofreading enzymes that can correct these errors. If the errors are missed, and the proofreading isn't 100%, so there are a few errors that are missed and do get passed on to the next generation. The result of these errors may be cell death, or it could trigger a disease such as cancer. And that's it for DNA replication. Go to my YouTube channel if you need some more help, or you can replay this.